I've learned from Sean Smith a little bit more about how the LinkedIn algorithm works, about how to get your posts seen. And in a future week, I and Sean will take this time to teach you how to do it. But for now, I'm just going to finish a word or two here and invite Lee, if you would kindly, you introduce me to our esteemed featured speaker today. Tell us, how do you know Doug Spitz and why you recommended him as a superlative speaker to educate us this fine Friday? You just need to come off mute first.
Probably as useful to hear me talk on mute.
There you go.
I'd also like to introduce Raul, who's part of the Inflammasense company that will probably be joining this group full time. This is his inaugural visit, so I can see Raul on the screen. So welcome, Raul. Doug Spitz and I met. He has a company in the iodine space. They are the world's, and his founding partner is the world's leading experts in iodine, and they work with molecular iodine. He'll go into more detail about it. The way that I met him is through one of the world-renowned infectious disease doctors. I had a chronic sinus problem for decades, and we decided to do an IRB N of 1 to see how to deal with it from what appears to be a bacteria-resistant, antibiotic-resistant bacteria. And this infectious disease expert had recommended that I use Doug's product. And I just thought that, and he could take his product and start dealing with implantable devices and so forth. So rather than me talk and not make a proper introduction, I'll just hand it over to Doug and let Doug introduce himself. But the infectious disease specialist had done research and written papers on this product, which is why they wanted me to experiment with it. I'm doing an IRB N of 1 on my sinus, and it's going to be with Doug's product. So with that, Doug, I'd like to turn it over to you. And Joe, I'm going to pop off for a bit, and I'll be back in a moment.
Okay, thank you for that introduction, and welcome to Doug. And go ahead, Doug, and tell us about your adventure.
Well, thank you for having me, Joe, and thank you for the intro, Lee. I am the CEO of I2 Pure, and we have been doing some very, very interesting things. It's been quite exciting. And so before I kind of get into that adventure, I need to do a iodine 101 class for about 5 minutes, so everyone completely understands where I'm going and what I'll be talking about.
Please do.
And there's a very good chance that everyone who listens to this, watches this, will have gone to the hospital or the doctor's office at one time and has had their skin cleaned with that red liquid known as betadine, povidone iodine. It is an iodophore. And when they clean your skin with that material, there is only 1 to 8 parts per million of molecular iodine. Molecular iodine is the only species of iodine that is microbicidal. It's the only part that's killing. When you look at the label, it says there's 1% available iodine, which is 10,000 parts per million. So you have a lot of iodine in that formulation that is adding to toxicity, that is adding to staining. The complexing agent is adding to acidity. And you have this very small amount that is doing the cleaning of the skin. And it's done a remarkable job for 200 years. What I2 Pure has done is we have unlocked that molecular iodine from its normal embodiments. And to put that into context, we have put ranges of this concentration of active into human skin and have done cellular viability testing with NAMSA as high as 15,000 parts per million. So let's just say about 3,000 times the concentration that the iodine that they used to clean your skin with, we put into human skin, and it had the same exact toxicity as the control, which was just 100% glycerin. So no toxicity whatsoever, no staining. And the very interesting thing is that when you put molecular iodine on human skin, it can be absorbed as deep as the hypodermis, and then it comes out of the skin as a room temperature vapor, and it is persistently biocidal. So we can turn the skin into this antimicrobial material, highly effective antimicrobial material, for 24 hours or longer. So we have turned this iodine into this molecular iodine into a platform technology. So when you take a look at the polymers that have multiple uses within medical devices, especially a lot of the interventional medical devices, such as catheters, such as grafts, such as sutures, the list goes on and on. Each polymer has its own degree of vapor porosity, and we are able to partition the molecular iodine into the polymer so that the gas will be able to escape from the polymer over many different periods of time. In some instances, we have some off-the-shelf polymers that we're able to imbibe, to partition the molecular iodine with for greater than 30 days. So you can imagine a catheter for the first time having microbicidal characteristics for longer than a month. And we are doing this across the entire spectrum of polymers, as well as creating bioabsorbable polymers that we're able to use as coatings. So we'll be able to coat things like orthopedic hardware and other non-porous titanium, hard plastic types of implants that will be utilized in the body. So we imagine an entire continuum of care, from skin prep through medical device, through wound irrigation, through closures, that will prevent the pathogens from entering any vector of interest that the pathogens have been taking to date, causing really what is the most troubling of surgical or interventional activities, and that's infection.
Doug, wow. Most of it went over my head, but what I did understand was very exciting and consistent with a couple of folks in my network that are working on things in your space. One of them is a scientist who came to our event last year working on something of this nature. Some time ago, I did a year with Teleflex as a marketing consultant on largely their cardiovascular and their catheters and the like. And I know that there was a great deal, I've forgotten a lot, but there was a great deal around antimicrobial features of their products. I do recall that there was something endemic in the Japanese society where a treatment, an antimicrobial treatment, had an adverse effect on that population. I don't remember much more than that. Help me understand, you've talked about the concentration and percentages and how much can be done. Help me understand how a hospital system or folks who would use your product would understand the delta between what you're working on and what is presently available in the marketplace.
So it's a great question, because when you take a look at what's in the market space today, and some of these products that they call antimicrobial, what they truly are is they are biostatic. And when you look at these products, whether they're catheters, what have you, what they are talking about in their literature is how clean that product is when it is retrieved from the body. So having a biostatic product doesn't allow a pathogen to grow on the surface of that product. That product many times you hear about silver, or you hear about copper, you hear about these ion coatings.
Mhm.
And these ion coatings, in some instances take 30.
He's temporarily frozen there. He comes back real fast. In the meanwhile, Gunjan, you've had your hand raised for some time. What did you want to contribute?
Is a microbicidal surface.
Doug, we lost you for a good 30 seconds there.
I am sorry. Am I back?
You are.
Okay, good. So these coatings that I was talking about are designed to keep pathogens from growing on them. So they still give a vector of attack for the pathogens to enter the body, but not take root on the product. Where our products will be able to destroy and kill pathogens directly. So they will not only be able to not take root on the device, but they will not be able to take root in the tissues that the device is being used to support.
I believe you said that when applied to a device, the antimicrobial features will extend beyond the product itself and be absorbable into the body?
Not absorbable into the body as much as making the tissues around that device microbicidal. So we're not.
Help me understand that. I got real close, and then you said microbicidal,
So the device has this partitioned iodine in it. It comes out as a gas. This gas is going to interact with the environment completely surrounding it.
Okay.
And so with regards to, let me just use this as an example. As regards to a catheter, especially people who have the chronic need for catheterization, many of them have chronic UTIs due to biofilms in the urethra.
Mm-hmm.
This is further down the line, but one of our thoughts is that someone who has a chronic UTI due to this biofilm, a catheter that has our iodine could be used as a therapy to destroy these biofilms, because that catheter will be against that tissue, impacting the tissues that surround that catheter.
I'm going to call on some folks because first, thank you so much for being here. Lee was right. And for me to be fascinated, I'm some marketing guy. Most of this is going through me. I can only imagine what David, who works at Cook, is thinking. Eddie is working on a catheter right now. Kevin has a lot of experience in the space as well. Gunjan's had his hand raised for a while. Rick has his hand raised. So we're going to use all the time today. I'm pretty confident. David?
Doug, sorry I missed the first few minutes, but can you tell me when your iodine agent, I guess I heard, goes into the tissues, if I remove the catheter, for example, what's happening in the tissues? Is your stuff still active?
Well, it's a good question. And I would have to say that we have not studied that. So I can't give you a direct answer. I can say that I would anticipate that there would be some degree of persistence. I don't know whether that's going to be minutes or hours, but the likelihood of persistence would be there.
So if I worked at the agency, probably around the world, I would advocate from my side as a regulator that you have a drug. You don't really have a device. You're not protecting the device. You're interacting in the tissue, and you require the tissue interaction for your response. This becomes definition of drug, not device. Devices, like, if I can just take a moment, if you took a vascular stent, a metal stent, prop the vessel open, it's a device. When you put drugs on it in a combination, the stent will keep the vessel open, but the drugs are there for the extra effect of interacting with the tissue, of preventing reinvagination and regrowth of the endothelium to block the vessel again. So I hear the same thing from you in your description, and unless you said something different in the very beginning when I was tied up.
I will say that to a certain, that you are correct with the definition of a drug. But we have 3 products that are going to be FDA approved this year, and they are all devices. And the only thing that we are doing in these devices is acting as a preservative for the device.
Interesting. I would just suggest you might look at what stays behind in the tissue and is it still active.
Doug, how long have you been working on this?
3 years.
Have
And-
you already had a pre-sub conversation with the FDA, and so that you know that they're going to see this as a
That's a great question.
device-led? What?
There are multiple predicate products with iodine in them already, and we are following the roadmap that has been set out before us to get products to market as soon as possible.
Doug, I would say, you recall when I engaged you just yesterday, the day before, I said this call is very much a family and we're all
Yeah.
helping one another. I strongly advise you to take a call with Michelle. She has something she starts everyone with called a regulatory pathway assessment. And going through that, just double check if it's a de novo, if it's all of the things. This is what she does.
And I very much welcome that, and I would love to do that. But I will tell you also that I2 Pure is a small company, and we do not have the regulatory expertise, the commercialization and distribution horsepower that we would want to put our products out with. So we are partnering with companies that are in the space, that have the requisite experience and horsepower to do this. But I am very interested in having this conversation with Michelle because our focus as a company is catheters and wound care. And we are allowing our partners to bring the dozens and dozens of other indications to market. So we're not down. We haven't even started the regulatory process for the 2 indications, which I had just mentioned. But we have probably 5 or 6 other products now that are in the pipeline, which one is under contract by a top 10 global life science company. So
A lot of the folks on the call don't have, just as you are smaller or starting up, they don't have all of the resources. That's why we talk to one another. I just put in the chat for everyone on the call a copy, I hope you don't mind, Michelle, of your regulatory pathway assessment form. Give it a look. It intimidates me, so put time on our calendar. Michelle, go ahead and, I will. I'll just put a link to her. You mind if I put a link to your team's calendar in the chat? Oh,
yeah.
you've already done it.
So,
Thank you.
so Joe, I just wanted to kind of
Right.
respond to Dave Chadwick's, question there, because I've supported registrations for both coated catheters and wound care for Bard, BD, Teleflex, 3M, all of these guys, and it really comes down, like a lot of things, to what claim you're making. So, again, if it's preservative in the product, it's pretty simple, but the minute you make any healthcare claim, it does become a combination device. And the approval pathway on those has become significantly more difficult over the last 10 years. Even if forget about, something, an insertion catheter, even for topicals, I've had a couple of projects kind of blow up on me on topicals because all of a sudden the agency is now requiring multi-site clinical trials, even for basically what should be a 510K.
Well, and anything with an antimicrobial claim, period, no matter where it's applied or what it's applied to. Like, I know surgical masks that were trying to make antimicrobial claims during COVID, the FDA wanted $500,000 worth of testing. Just for something as simple as a surgical mask, much less an implant. So
I've got a question.
Hold on just a sec, please. It's 3.5 weeks away. Maybe it's too tight. Most of the people on this call will be meeting with me in person in San Diego on the 11th, 12th, and 13th at our 10X Medical Device Conference. I don't mind doing a quick commercial and telling you it's the strongest faculty I've ever had. It'd be great if you could come. Now, Larry, you.
Yeah, I got a question. Yeah, question on this is important from the standpoint of your future marketplace. What is the clinical benefit of this? In other words, I assume it's like reducing post-procedural infections, where we know when a catheterization is done, there's always a potential for introducing bacteria. And the consummate study would be a comparison to products without your coating and with your coating, and showing that the infection rate in your patients is lower, post-procedural infection rate. Now, that study may be easy to do if the post-procedural infection is high, but if the post-procedural infection is relatively low on all of them, showing a diminishing of that may be difficult. But ultimately, if I'm a doctor or I'm a hospital and I see there's this new product and maybe there's a little bit of a premium in its price, I'm going to say, what's this going to buy us? How is this going to help our patients? What is the clinical outcomes that are going to be improved by it? So it's something down the road, but it's something you should be thinking about.
Well, I will tell you, I'm very excited that one of the, we think we're going to have significant impact with one of the products that's coming to market, which is a dermal regeneration matrix for full thickness wounds for burns and for trauma. Because currently in that space, they have these scaffoldings that they put into these wounds, and these scaffoldings are just exquisite at growing dermal cells and cells in general. But unfortunately, when you're great at growing cells, you're great at growing bacteria and fungus. And many times there are infections with one of the areas of the burn that have to go into, the patient needs to go back into the OR and debride that material out of the wound and then reapply and start all over. And we believe that we are going to have a significant impact on the infections within those burn treatment centers and trauma centers for dermal regrowth.
Rick?
Just unmuting. I've got, Doug, this is frankly amazing. I was raised by my father, who's PhD in microbiology, and so a lot of this stuff just got osmosis transferred to me, but this is absolutely amazing. So I've got one question that's really in 3 parts. And this is in the hypothetical instance that I'm designing a long-term implantable device composed primarily of PEEK, that is long-term in terms of more than 29 days. The first question, I'll just say them all together and then run them because context is important. So the first one will be, what is the design pathway to use the iodine product in this product I'm mentioning? And the second one would be, what's the manufacturing process for applying the coating? And the third would be, as far as approvals go, in case of a near convergence, if this product is ready a little early, what are the specification preparations that I can make to most easily add this into the product at a slightly later date when you get release? So the first question was, what's the design pathway, or what design allowances do I need to make to be able to use a product like this with a PEEK long-term implantable?
So I can't give you a wholesale answer on that without so there are multiple pathways of manufacturing, because there are different ways in which we partition the iodine into a device. So it may be such that we are taking the polymers and partitioning the polymers pre-manufacturing, so it becomes part of the manufacturing process. Or it could be post-manufacturing, where we're partitioning the iodine into the completed device, and then it will give us a different type of challenge as it relates to shelf stability. Because ultimately this is all about, you need a product that is shelf stable for a couple of years.
Uh-oh, we've got you.
Okay.
Doug, you're temporarily frozen, still frozen, and I don't want to miss anything you're saying.
The distribution process, we would be able to.
Douglas, I need you to rewind another 20 seconds. We lost you there.
Okay, I apologize. The bottom line is there are multiple manufacturing processes, and there are multiple processes to partition the iodine into these devices. So we will have to figure out, is it a pre-manufacturing situation, or is it a post-manufacturing situation where we would partition the iodine into your device to make it a shelf-stable device that could sit on the shelf for 2 years and be used and have the iodine viable.
Okay, so it sounds like question number 3 really depends on the answer to the first 2, and sounds like that'll be a phone call right after I get the numbers on toxicity and infection with similar devices. So and then we'll take a look at the risk and risk rewards.
And I'll tell you that with the dermal regeneration matrix for the FDA 510K, it required a pig healing study, which required, I believe, a 14, 28, and 90-day study. And we had to, so the matrix that is currently going to be part of this solution is a matrix that currently has a 510K approval already. So we had to prove that we did not impact the healing, as well as and this is a bioabsorbable pad. So histology comes back, and I will tell you the histology, it couldn't have been any better. So we're very, very excited. When it comes to iodine, and I talked about it not being toxic, and it's so compatible to human tissue, it's very important to understand that every vertebrate on the planet has an iodine cycle. That the oldest living organism on the planet, with fossils dating back 3.5 billion years, being blue-green algae, has an iodine cycle, and that our own thyroid manufactures molecular iodine in concentrations as high as 720 ppm. Our bodies then turn that molecular iodine into a hypoiodious acid. It's bound with thyroglobulin. Then you have thyroid hormone.
This, to somebody with even the least amount of knowledge about this, which is me, this is fantastic. This is just really amazing. So throw that in and mute myself again.
Well,
Eddie, you've been very
thank you
patient.
very much.
So Eddie is working on a catheter right now. Eddie, a quick plug for what you're working with.
Hi there. Yeah, mine is an indwelling catheter that's designed actually to stay completely inside the body. And our design is actually to have the urethra close over the end of the catheter, so working with the body's own natural defense mechanisms. In fact, just a couple of days ago, I was talking with our medical director, and we're debating the idea of coatings. And aside from what Michelle mentioned, which is it would completely mess up our regulatory pathway from where we are at the moment. He said, look, our whole claim is that we're working with the body's natural defenses. If we put a coating on it, wouldn't that kind of undermine our basic premise of the way we're approaching things? More significantly, I just came back from the EAU, the European Association of Urology conference, where I spent a half a day all about infection issues. And it's something I have another history in. I, however, am not a scientist, for a long, long time, and I'm not an expert in this area. But the key issue is that most antibiotics, and I understand that iodine not an antibiotic, but most of these things have shown that there's resistance that develops in quite a short period of time, 3 years. Now, 3 years sounds short to us, but actually, if you look at it from the bacterial's life cycle point of view, it's equivalent, 3 years is equivalent to about 80,000 years of human evolution, right? And we look pretty different than we did 80,000 years ago. And if you look at 70 years, if you look at 70 years of antibiotic usage, that's why we have the resistance problems that we do. Likewise, the other thing I'm just going to throw out there is that with any of these things like silver coatings, again, there has not been any demonstrable long-term benefit shown to silver coatings. And I think of iodine more in that category, silver, copper, and things like that. So those are the cautionary things I would mention, but I'm always interested and intrigued to learn more.
Well, one, there has never been any proven resistance to iodine from any pathogen. Nothing seems to be resistant to it. When it was first discovered and the first embodiment, which was Lugol's, formulated in 1829, it was designed to fight against tuberculosis, which of course they didn't have a good delivery system for that at the time. However, after almost 200 years, Mycobacterium are still not resistant to iodine. As it relates to the other coatings that you mentioned, again, they fall into the biostatic realm. There are no coatings on any medical devices that are biocidal. And those coatings are biostatic. So I can tell you with the flow of blood, the flow of medicine, or the flow of urine, it is going to be much quicker than the average of 15 minutes that's going to go across that surface. And with a fluid, an exudate, a medication, what have you, you then also have the bioload that is protecting those additional pathogens from that biostatic coating. So that's why the clinical benefit of biostatic coatings has been so just not, the bang for the buck hasn't been there with the biostatic coatings. There's a reason and I'm not going to go into the science of the cellular activity, the molecular activity of the of molecular iodine, but from a efficacy perspective, at 1500 ppm, when you start looking at these efficacy studies and when they start talking about high-level disinfection, all high-level disinfection and sterilization is greater than 6 log. Well, every microbe that we've been put up against in third-party testing, we've been greater than a 6 log reduction within 30 seconds. So we feel quite comfortable that the material will be quite advantageous in many, many different embodiments.
But did you say, I mean, that all sounds really, really encouraging, but did you say that you don't know what the mechanism of action is?
Oh, we know exactly what the mechanism of action is.
Oh, you do?
Yeah.
Great. That's my jam. I love understanding that, because if you understand that, then you know why it's working. But a lot of people don't understand their mechanisms of action.
So not only do we understand the mechanism of action, but here's going with this group, because we're talking about devices, we're not talking about topicals, But when we did this NAMSA study with multiple concentrations from 500 ppm up to 15,000 ppm, we also demonstrated in a secondary study a dose response to the iodine being put into the skin that was generating anti-inflammatory and anti-proliferative cytokines. In July of this year, we're doing human trials for chronic wounds down in the Caribbean, because in our pilots, we have demonstrated that in some instances, we're healing wounds 80% faster than docs had ever seen before. When the wounds were healing, there was limited to no scarring, and it was a very strong anti-inflammatory. And there have been, when we noticed these characteristics, which we noticed early on a few years ago, we started looking for research that was kind of pointing out some of these characteristics in older iodine embodiments. And there was a very interesting medical manuscript published by a PhD by the name of Yuri Wormser that used live pigs and scalded the skin and allowed the skin to stay in contact with 75 degrees C water for 10 seconds. And the first test, untreated, of course, the skin got ulcerated and was very, very angry. Then he did it again and he treated the skin after the burn, and the skin, although there was hyperplasia, there was no ulceration. And then he pre-treated the skin. No ulceration, no hyperplasia. The skin was not happy, but it was intact. So not only does the molecular iodine have this microbicidal characteristics and these healing characteristics, but it also has a protective characteristic as well.
Interesting. And that actually, as you were just describing all this, going back to the whole point of 1829, I think you mentioned, where are you on the IP horizon?
So we have one granted patent. We have 2 patent applications submitted, and we have 4 additional provisionals that will be submitted over the next 12 months.
Great. John Richard?
Yeah, I was having a little trouble with my mute switch. People tell me that's a real problem with most of my life. But okay. I was thinking about the approval mechanisms, and I was thinking about your device. And I was wondering, and I don't know if Michelle is still here, but yes, she is. Okay. And might have something to say about this, but I wonder if there's a difference in terms of, there was a concern that if we're adding the iodine coating or infusion, or however it's put in this case, to the device, it might contradict the device's claim of being, basically infection-free. I'm not sure what the best term is. So my question was, in light of that, is there a separate category for installation inflammation or installation trauma versus claims of long-term biological tolerance and infection resistance? Would that be, Ed, possibly a way to deal with that? Talking about the iodine is taking care of any installation trauma, and then of course you've got a different situation after it's been in place for a while.
Frankly, I'm not sure. I would defer to somebody like Michelle or others on that sort of thing from a regulatory, what can we claim point of view.
A comment. I think the basic issue is, is the product can be used more safely, whether it's short-term or long-term. The key is clinical outcomes. Can you show that when a product is used that has this coating, there's less infections? And that's the key issue I think that you're going to face with FDA is, what is the clinical utility of this? It's theoretical. You said you're going to do a study in Central America, I guess, but the key is understanding the mechanism is clearly important, but what is the clinical utility in the standpoint of short-term or long-term? It's just, well, how does the patient benefit? And that's obviously a great opportunity for you from a market standpoint, but also FDA is going to look at that.
Larry is an expert witness for FDA. He's also a helpful person to know.
So Doug, I really like the discussion. I like where I think your product can go. I must admit, I was sitting here running through your website, and your website says something like, quoting one of your papers, which is what FDA will read. Once applied, molecular iodine is absorbed into the epidermis, dermis, and subcutaneous tissue, where it becomes a gas and is released through the skin. Can you help me understand what that means in the claim world, the promotion world that you'll eventually get to?
So when iodine is put into the skin, it gets partitioned into what we're thinking is near lipid material. The fact that iodine, which is highly hydrophobic, finds the skin a comfortable place to go is still a bit of a mystery, but yeah. Hey, it's there. But a large majority of the iodine is then, it's partitioned into the dermis, and then it comes out of the dermis as a gas. So it's coming back, it comes back out. Now, the preeminent dermal absorption studies were done in 1982 by Dr. Braverman, who through mucosal tissues, so, not dermis, but mucosa, demonstrated that he could have a 5% permeability utilizing PVPI. The benefit of the iodine is that of course, the FDA, when it comes time for us to deliver these types of therapeutics and go down the NDA/IND route, the thing they're going to be concerned about with iodine is thyroid toxicity. Mm-hmm. And the thyroid is designed to pick up the iodide, not iodine. And we have safety data on dosing nearly 3,000 patients with very, very high levels of molecular iodine with no adverse events.
So you just made a comment that when you go down the IND, NDA road with FDA, which is the drug side of the house.
Yes.
So you're really thinking this has drug action, treat, prevent, mitigate, just straight off the definition.
Well, we cure toenail fungus. We cure, we cure just about every topical infection there is, and that is a drug.
Yes, it is. Okay, good. Thanks. So you're going to end up maybe with a combination when you come to the device side.
Correct.
Drug house driven combination. Fine.
But many of the devices will be purely just devices, sure, because we will not be making any antimicrobial claims.
Good. Thank you. Good clarification for me.
Lee, you've returned. And you're on mute.
Yes, I have returned.
Well, you were right. Superlative guest. Now that we understand what he's working on a bit more, how are you two working together, if at all?
Well, we're probably going to be using his product on my sinuses. The thought is that I've had a chronic sinus problem about every 30 months, plus or minus something, since I'm about 7. I've had some problem, a flare up. In between, I've had a consistent postnasal drip now for 60 years plus. And they did surgery on me when I was in 2nd grade and took out my tonsils. They did sinus surgery on me. They dealt with my eardrums in order to get airflow. Nothing seems to have worked. I've always been able to shake it. Antibiotics never worked, although they threw lots of antibiotics at me over the years. And as I've gotten older, I don't know whether you can tell I'm a bit older than most of the
I
people.
noticed.
Oh, okay. Thanks for reminding me. That this last time, it was very hard for me to shake it when it flared up. I like sort of went for 14 weeks with me coughing, not 24 hours a day, but some portion of every one of the 24 hours where I was having a coughing. Eventually, we were able to get it under control. But now I wanted to find out, the thought was that it might be an antibiotic resistant bacteria like Staph aureus that, like, for people who have eczema, flares periodically, or like some of the other diseases that people get that flare
Now, Lee,
periodically.
you know that this call is not HIPAA protected, right?
That's fine.
Yeah.
Are
you working with him in a more?
I'm not working.
Are you working with him also, not only as a patient, but in a more collaborative way for projects you're working on?
At this stage, my main focus is to put together the principal investigators. It's going to require an allergist, an ENT, and an infectious disease physician. And but they're all identified. We were putting that together. The protocol's being written up, and we're submitting it to IRB. I've talked periodically. Some people on the call have heard me talk about it. I'm a big fan of N of 1 IRB research. I've done a number of them. While they're statistically irrelevant, they're not irrelevant to the person who's the one. And in this case, I'm going to be my own N of 1. But because I'm playing around with serious stuff here, I want to make sure that it's an IRB protocol is followed. So I'm just not going out there on some like renegade sort of try stuff that in ways that shouldn't be tried. So we'll be doing before procedure scans, during procedure swabs, during procedure swabs. There's MRIs being built into the protocol because there is some inflammation in there. And so that's my main use. There is a deck floating around because he's doing a round right now. I'm trying not to commingle those two things. As a patient, I want the IRB to be clean. I like to go through. I like to see if this is the thing that does cure my chronic sinus problem. And if it is, that's wonderful for me and probably for the IRBs that'll follow me because of my success. If it isn't, well, then I don't need to think about commingling investment or other evaluation.
Larry, I'm just curious if you've heard anything from an FDA standpoint, or you were to be put on the stand one day about what you've heard today. You're still on mute. Any kind of things you might bring to Doug's attention, make sure you this or that.
Well, no, the one thing that is positive is he talked about the predicate devices that apparently iodine coating has been used on other products. And if it is, then that stands with FDA as a basis of safety if they've not had a lot of adverse reports upon it. The key is gaining the FDA authorization to sell is a great protection in product liability cases. The opposite is true. Putting out a device that has not gone through FDA and having a significant problem occur with it to the point where somebody's injured and sues, you have no defense. Put me on a stand and I'll tell everybody you're a bad person.
Hold on for just 1 second.
I think he's getting someone to battle you or something. I'm not entirely sure.
As far as just, our claims of non-toxicity and my concern of future adverse events, I travel with 1500 ppm. I'm used to the taste, but the amount.
This proves nothing. You built up an immunity, just like Wesley in Princess Bride.
Well, I will tell you that
was iocane powder, wasn't it?
This was 1500 ppm of molecular iodine.
Inconceivable.
Very well done.
Thanks.
I will tell you that the US recommended daily allowance for iodine is 150 micrograms. I'll tell you that the upper limit that they put is about 1,000. And there are many prefectures in Japan where are in that blue zone, where those people in that blue zone are excreting 10,000 micrograms of iodine a day.
I hope we wouldn't get to excreting on this call. Speaking of excretions, Eddie, you had something to say.
Yes, Lee, only because you told me you've suffered a lifetime of sinus problems, I thought I would share a clinical tidbit that you are unlikely to hear from any physician, and that is how to apply anatomy knowledge to help accelerate
Dr.
healing.
Lin is an anesthesiologist.
But I'm not past any gas when I'm here.
Go on.
All right. So when you look at the sinus, the maxillary sinus is our biggest sinus, but the opening is at the very top. So think about the sinus as being almost, it's a big cavity, and it doesn't drain until it fills up to the top and it overflows. But anatomically, you can reverse it. So the opening is at the bottom by tipping your head down, bend over until your head is pointing towards the floor. And if you sniffle really hard, you will cause a Venturi effect by the rapid air flowing past the opening called the os, and it will suck out the tenacious mucus and other purulent material that's in there. So you can really accelerate the draining. And if your infection is on the right side, when you sleep at night, lie on your left side, because then it's in the upper portion and it will drain out. And you can reverse for the other side. And severe coughing after a viral illness or sinus infection is caused by hyperreactive airway. And oftentimes, and I have that, I mean, I would have severe coughing for almost 2 weeks after I have recovered fully. And that can be treated by a steroid inhaler, and that would tamp that coughing spell down very quickly. I have a friend who recovered from COVID, and for almost 2 weeks, and he's been seeing doctors, he would cough so hard that he starts vomiting. So when I recommended that treatment to him, that same night, all the coughing went away.
I will,
Thank you.
the one thing I will say about chronic wounds and chronic disease, many times, there are one of two, if not both, characteristics present. One is generally a biofilm, and the other is chronic inflammation. And one of the things that I'm feeling very positive about a good outcome with Lee will be the fact that we can destroy both.
That
While
would be
keeping
great.
Lee intact.
While keeping Lee intact.
Okay. Good.
And that may or may not be a good thing. But
I think it's a good thing.
But anyway, just they had me on a variety of steroid inhalers. They had me on a variety of steroids. They had me on a variety of steroid nasals. I have the yoga inversion, not that teeter thing, but yoga inversion, where I go 180 degrees upside down to be dealing with that.
As your moderator, I'm going to gently guide us away from more stories about your nostrils.
Anyway, I
would like to
thank
hear-
you very much, and Doug, thank you very much for presenting today. And
Well, thank you for having me.
Before you run, I'd like to get closing thoughts from Eddie and from Kevin. Eddie, do you see potential in working with Doug in some capacity? You're on mute. No, Ed Capabianca.
Yeah, there's a problem of two Eds on the call.
And I both affectionately call you Eddie.
Possibly, we are exploring potential for using coatings on our catheters. So we're at an early stage on all of that. It's quite possible, like say, that you can explain to me the mechanism of action is also very, very helpful. So yeah, let's we can continue it on offline.
Very good.
And Kevin, I'm interested in what you've heard relative to all the things.
Yeah, so for some applications, we may be in competition, although I think based on kind of the direction that we're going, finished product wise, probably not. I would say we're maybe competitors in all of the theoretical applications, but commercially, I know we're probably not. And I was actually going to reach out to Doug after the call because I actually do have some ideas for him. And I would be interested in, and Doug, just so you know, my background for the last 25 years, I've been very involved, particularly with chlorhexidine, but also with PVP iodine, selling into those markets. So I definitely have some thoughts for you.
Wonderful.
And I actually, I'll send you a calendar link for next week because I have to go through my notes from pre-COVID. I need to see if I can resurrect a contact from years and years ago who had reached back out to me with a product idea right before COVID hit. And it might be interesting. And more importantly for you, they have an awful lot of money to spend. So we can maybe chat next week.
Wonderful. And I put my email address in the chat. So anyone on the panel who wants my email address can get it there in the webinar chat.
Thank you. And to my eye, it looks as though 6 people presently on screen will be with me having ice cream in a few weeks. Doug, any chance that you might be able to come out?
Possibly. I was out there for NextMed.
Okay. Oh, great. I love Daniel. How was did you stay throughout? It closed yesterday?
I stayed through, so it closed yesterday and I left yesterday. I left yesterday first thing in the morning. So I was there all 3 days.
Mm-hmm.
Daniel's great. It was probably one of the best conferences I've ever been to.
That's how I felt when I went. Tell me, is that your first time with him?
That's my first time with them.
Okay.
And I found the group to be remarkably collaborative. You know, it's just a very open group. It was certainly not an East Coast affair.
The first time I went, I felt euphoric, and it was akin to being at a Wharton reunion because I was around other people who were all kind of at that level. Okay. David Chatwick, I don't mind sharing this with the group because I often solicit from the group what we should be talking about. You say you may have a potential topic for a future week.
So yes, I got to go back and find my thing. So last week I was on a webinar hosted by a law firm. And the title was, I thought interesting for this group, or for, the composition changes sometimes, online tracking technologies. What Google and Facebook does with when we search, foot care or fingernails. Online tracking technologies, new HIPAA and FTC guidance and actions change the enforcement landscape. So these attorneys who, if you thought it was interesting, I'd be certainly willing to reach out to them and see if they would chat with us, or one of them. There have been probable HIPAA violations in the tracking data. They had an example of, you go to, just to pick a company, GoodRx, who did get a lawsuit against them. They lost it. And I'm making this up, but I go in there to GoodRx and I order my toenail fungus, Doug mentioned, cream. The Metas of the world have contracts to capture this from the GoodRx firm. Is it de-identified? Is it not? Should we care? Should some of these people in the circle that you have on my screen care, more about this kind of tracking, and what does it mean, and what's the regulatory landscape looking like? I cared about Amazon knowing what I search, but I never felt it was going to come into my medical world.
I wonder what HIPAA implications there are.
They're big.
And I'm sure that they're just too smart as a company to do anything that's a direct violation of something, as I would hope.
But who knows? It's just an avenue. There's been a the office. I learned this. I didn't know they existed. The Office of Civil Rights, which is part of Health and Human Services, has filed action against them.
Yeah, I personally would find that interesting. If you have a connection there, by all means, recommend them.
And I can reach out to one of the lawyers and connect you to.
Okay.
See if you want to develop it into something. But
sure,
touching each of us in the healthcare world and what we say and what we do, and especially entrepreneurial guys and girls here, would have a big effect. And it's an area of regulatory I've never worried about before.
Thank you, David.
Open my
eyes. Doug, what a pleasure.
Yeah.
Yes,
thank you.
And before you leave, I want to show, if you're interested, I can show you the most recent thing.
Oh, that's another 20. Okay, go ahead.
One of our attendees actually created part of my lab.
Please.
So let me share this. Let me share the screen.
Some folks may jump off. We've gone over a bit, but I'm recording.
That's fine.
Please do.
Because what you're looking at is a self-adhesive
So far,
iodine.
we're not looking at it.
You don't see it?
Not yet. Here we go.
You're looking at a self-adhesive iodine hydrogel bandage.
How do you apply that? Do you pour
You
it onto your hand and wait for it to kind of harden?
No. The bandage it would be solid. It would be a solid that you would just slap on a
Take out
burn.
of,
You would take it out of a Teflon package. And
I'm sorry, my packaging person isn't on. I'm curious, is it like wrapped like a traditional Band-Aid from Johnson & Johnson?
it's not wrapped. The bandage itself was just created last week.
Okay,
That was
Well-
a demo, Joe. That was a demo.
okay, fair
No
enough. And
problem.
Douglas, I'm sure you know, but I feel compelled to say the one thing that Jan taught me that I never forgot, and that is that packaging in medical devices is the medical device. It's not the thing it comes in, it's part of it and is treated that way. So if you don't have a packaging engineer or expert on your team
Yeah.
and you're going down that path, I know a gal.
Yeah, well, you're right. It's very important. And lots of interesting things that we're doing in that space as well.
Normally I would say thanks everybody and goodbye, but I'm gonna ask Doug, stay another couple minutes for me. Anyone who wants to leave can. What were some of your biggest takeaways from NextMed and Daniel Kraft's event? Bye, Eddie. Douglas, we're losing your connectivity. It's really getting spottier.
Yeah, the network,
Maybe it wasn't meant to be.
you know, are you there? Do you
Yeah,
hear me?
kind of.
The conference was very focused on the future of data collection and the future of big data, AI,
Mhm.
and how it's going to be used in the future of healthcare. Since healthcare is becoming more and more self-service than it is the
Right,
old model where
right.
it was a high touch model.
How many guests were there?
About 300. So there were a lot of conflicting conferences going on at the same time, which I think hurt Daniel.
Yeah.
And I will say that the conference was a bit light on where the rubber
Where hits the road is the end of the sentence.
healing.
Mm-hmm.
But on and this global community-based health improvement that is this journey. You had some practicing MDs who take care of it's all about democratizing medicine, making community healthcare not only more accessible but more effective for the individual.
This was his first event outside of Singularity University. Were Ray Kurzweil or Peter Diamandis in attendance, either virtually or in person?
I'm sorry, you broke up.
No, neither Ray or Peter were there.
Okay.
I
right,
was
well-
there on behalf of X Prize. Anusha came, who's
Okay.
the CEO of X Prize. And we were also very active in helping market and try to support what he was doing. So,
Okay. Well, thank you. And I will close here. Douglas, profound thanks. I greatly hope you get to meet you in person in a few weeks. And Kevin and Rick, I will see you in a few weeks. Thank you, everyone.
Hey,
Have a good
Joe,
weekend.
I'm not 100% sure. I may have to be in Dubai that week. I should know next week.
I can't compete with Dubai, especially if somebody's paying to get you out there.
Hopefully. We'll
Yeah.
see.
All right.
I'll
Bye,
let you know
folks.
next week. Thanks.